All right, let's get started with translation. So we have our mRNA is all processed, it's spliced up, it's exported to the cytoplasm, it's ready to go. So we're going to first look in this screencast about the two other types of RNA that are involved in translation, which is tRNA and the ribosomal RNA in ribosomes. All right. So genetic information flows from mRNA to protein through the process of translation. Remember, that's our second half of our dogma. DNA, RNA, protein. Okay, so we're going to look at this step right here, RNA to protein. All right, so a cell translates an mRNA message into protein with the help of another type of RNA called tRNA. Okay, tRNAs transfer amino acids to the ribosome and put them in the right order in the growing polypeptide or protein. Okay, translation is a pretty complex process, but we're going to try to explain it as simply as we can, and it's important to understand all the different components before we move on to how the process actually works. So let's look at tRNA. tRNAs are all different. It's not like you have one kind of tRNA. You have many different kinds of tRNAs, okay? Each tRNA carries a specific amino acid on one end, okay? And the other end has an anticodon, okay? And the anticodon is complementary and can base pair with the codons on the mRNA, all right? So tRNAs, a little bit more about tRNAs, and then we'll show kind of how the whole mRNA, tRNA matches up. tRNAs, a single RNA strand that's only about 80 nucleotides long, okay? And if you flatten it, we're going to look at the slide, it looks like a clover leaf, but it's actually kind of L-shaped. So here's a tRNA. This is that clover leaf you're talking about if it gets flattened out. But you see it's just one strand, and it has this kind of foldy up process here. But if you look on that strand, there's a lot of complementary base pairing that gives it its 3D structure, okay? And then on one end, we have the site where the amino acid attaches. And on the other end, we have our anticodon. And again, this is complementary to the codon in the mRNA. 3D looks a little bit more like this L shape because of how it's bent up. And this is the kind of cartoon that we use to represent a tRNA. And there's the codon. The anticodon, sorry. Okay. Again, because of hydrogen bondings, tRNA twists and folds in a three-dimensional shape that is roughly L-shaped. All right. So accurate translation depends on two things, two steps that involve our tRNA. Okay. First of all, we have that site where we're going to attach the amino acid, but you have to attach the right amino acid. Okay, so a correct match between a tRNA and the amino acid, and this is done by an enzyme called aminoacyl tRNA synthetase. Okay, so that is done, the matching up of the tRNA and the amino acid. Second, you have to have a correct match between the tRNA anticodon and the mRNA codon, and that's done by base pairing. Okay, now remember we talked about that third base and how some of the amino acids have 4 codons or 6 codons, so that third base doesn't really matter. And how it kind of fits into protein synthesis itself is the third base exhibits this thing called wobble. So more than one, as long as the first 2 bases can base pair properly, that third base doesn't always matter, and it has to do with the redundancy in the code. So I'll talk about wobble more in class and in the next screencast. But it has to do with the redundancy in the code, and the fact that more than one codon codes for each amino acid. This is just showing you how tRNAs are assembled. So here's my enzyme that does it. We get the amino acid goes into an active site in the enzyme. Of course, ATP is required. Then the tRNA is put in, and the two are attached by the enzyme. And now we have our tRNA with the amino acid attached, and we call this a charged tRNA. So when it did not attach, it's uncharged. When it is attached, it's charged. All right, so that's our tRNAs. Now we're going to look at our ribosomes. And ribosomes is this big, giant complex of a lot of proteins, most of which are enzymes, and ribosomal RNA. Okay, so ribosomes, site of protein synthesis, site of translation, they facilitate the tRNA anticodons coupling with the mRNA codons. Okay, there are two subunits to a ribosome, the large and the small, and they're made of proteins plus rRNA. Okay, bacterial and eukaryotic ribosomes have some similarities, some differences, but there are some ways to specifically target bacterial ribosomes, and that's what most antibiotics do. They halt the bacterial ribosomes without harming eukaryotic ribosomes. So inside the ribosome, there are three binding sites for tRNA. There's the P site, and we'll see a cartoon of this on the next slide. P site holds the tRNA that carries the protein or the polypeptide chain. The A site holds the tRNA that's going to have the next protein or the next amino acid to be added to the chain. And then there's the E site, which is the exit site. And this is where once the tRNA discharges its amino acid and adds it to the protein chain, it's where the uncharged tRNAs leave the ribosome. So let's look and see those sites again here. So here I have my P site that holds the protein. My A site will have a tRNA that has the next amino acid. Okay, and the E site is the exit site. All right. So this is kind of the different binding sites. Here's my small subunit and my large subunit. The small one is small, the large one is large. And now let's look at the subunit with some tRNAs in place to show you how it works. So in the P site here, this is my P site, we've got the tRNA that's holding the growing polypeptide. What will happen is then into the A site, the next tRNA comes in with the next amino acid, and this polypeptide chain will be added onto that amino acid, and it'll be next in line. Once the polypeptide chain is cut off of this tRNA in the P site, it moves over to the E site, okay, and exits. See, now there's nothing there. It exits the ribosome, everything shifts, and then the A site opens up again. Okay, and that's basically translation. So you'll see in the next screencast. And then the last thing we want to talk about on this slideshow is just kind of how the whole DNA, mRNA, tRNA amino acid thing all fits in together. So I thought this slide showed it pretty well. So we have our template strand of DNA, and this is going to run totally wrong. Our template strand runs 3' to 5'. Our mRNA is synthesized 5' to 3' from the template strand, and you'll see it's complementary. Okay, so T would base pair with A with U, etc., and so on. Okay. Then each group of 3 here is a codon. So this AUG, GGA, GUU. And then for my tRNA, the anticodons are complementary to the codons in my mRNA. So you see A is complementary to U, U with A, G with C, that's 1. And then this AUG, as we know, is the start codon. It codes for methionine. So the methionine would be attached to the tRNA carrying this anticodon. GGA, okay, codes for glycine. Okay, So glycine is attached to a tRNA carrying the anticodon CCU, etc., and so on down the line. Now, I want you to just take a look here at some similarities, okay? So the mRNA is complementary to the template strand. But if you look, the mRNA is basically the same as the non-template strand. Okay? And while the tRNA is complementary to the mRNA, the tRNA is the same as, this is getting very complex, it's the same as the template strand. Okay? So just think about it like it's every other one is complementary the same, complementary the same. Okay? All right, hopefully that didn't confuse you, but you can kind of use these precepts to kind of move back and forth. And that's all. Okay, next screencast is translation in more detail, so go get that one.